welcome to Medical Dialogues, your daily dose of health and medical news. I'm Mr. Zaman and here is what I bring to you all from the world of medicine. How stem cells help in predicting a potential drug for polycystic kidney disease. In a new study in cell stem cells, scientists from the USC laboratory generated simple kidney-like structures called organoids and used them to identify potential drugs to treat adult-onset polycystic kidney disease. Polycystic kidney disease presents an autosomal dominant pattern of inheritance, meaning that the disease develops when a person inherits a bad copy of the gene PKD1 or PKD2 and the activity of the second good copy is also lost. Autosomal dominant polycystic kidney disease or ADPKD causes large fluid-filled cysts in many regions of the kidney, leading to the loss of kidney function and other life-threatening complications affecting the liver, pancreas, and heart. Tolvaptan, the only FDA-approved drug to treat autosomal dominant polycystic kidney disease, slows but does not block disease progression and only works in a subset of cysts composed of a particular kidney cell type. The scientists demonstrated that the organoids contained many of the cellular precursors and genetic signatures required to build the kidney during embryonic development. When implanted into a mouse, the organoids' nephron-like structures started to develop vasculature and even attained a limited capacity to filter waste, one of the most important functions of the kidney. To make the organoids useful for studying autosomal dominant polycystic kidney disease, the scientists used CRISPR/Cas9 gene editing to inactivate PKD1 or PKD2. As anticipated, the gene-edited organoids began to form cysts, which eventually detached and grew to centimeters in diameter. The scientists then performed the first screen using gene-edited human organoids to identify potential therapeutic drugs for ADPKD, focusing on a collection of enzyme inhibitors to give broad insight into the cellular mechanisms controlling cyst formation. After testing a collection of 247 enzyme inhibitor compounds on the organoids, scientists found 9 that inhibited the growth of the cysts without stunting the overall growth of the organoids. One compound, quinazoline, was particularly effective. In the future, organoids will become an increasingly powerful tool for modeling and understanding human disease, identifying potential treatments, and eventually providing transplants to replace organ function for patients, the researchers concluded. How smokers who smoke and wear masks are more prone to its harmful effects. Smoking traditional or non-combustible cigarettes while wearing a surgical mask results in a twofold rise in exhaled carbon monoxide and impaired blood vessel function compared to non-mask periods. That's the finding of research published in the European Journal of Preventing Cardiology or Journal of the ESC. The study focused on traditional, that is combustible cigarettes and non-combustible cigarettes, also called heat not burn or heated tobacco products. The researchers investigated the levels of exhaled carbon monoxide in smokers while wearing a mask during working hours and compared it to carbon monoxide levels during days off without a mask. In a second step, the researchers examined whether the change in carbon monoxide exposure was accompanied by impaired blood vessel function. The study included 40 smokers of conventional cigarettes, 40 exclusive heat not burn cigarette users, and 40 non-smokers with similar age and sex who were medical personnel in a university hospital. Individuals with known cardiovascular disease, hypertension, diabetes, dyslipidemia, chronic kidney disease, or atrial fibrillation were excluded as these conditions may affect vascular function. A similar number of combustible or non-combustible cigarettes were smoking during mask and non-mask periods. In conventional cigarette smokers, exhaled carbon monoxide increased from 8 parts per million at baseline to 12.15 parts per million with no mask and 17.45 parts per million with a mask. In non-combustible cigarette smokers, exhaled carbon monoxide increased from 1.15 parts per million at baseline to 1.43 parts per million with no mask and 2.20 parts per million with a mask. Among non-smokers, exhaled carbon monoxide did not differ between the baseline, non-mask and mask periods. In both combustible and non-combustible cigarette smokers, all vascular markers were higher while wearing a mask compared to the no mask. In non-smokers, there were no differences in vascular markers between the 3 periods, the researchers concluded. How salt intake is riskier, causing early death in people. People who add extra salt to their food at the table are at a higher risk of dying prematurely from any cause, according to a study of more than 5 lakh people published in the European Heart Journal. Compared to those who never or rarely added salt, those who always added salt to their food had a 28% increased risk of dying prematurely. In the general population, about 3 in every 100 people aged between 40 and 69 die prematurely. The increased risk from always adding salt to food seen in the current study suggests that 1 more person in every 100 may die prematurely in this age group. In addition, the study found a lower life expectancy among people who always added salt compared to those who never or rarely added salt. At the age of 50, 1.5 years and 2.28 years were knocked off the life expectancy of women and men, respectively, who always added salt to their food compared to those who never or rarely did. The study is the first to assess the relation between adding salt to foods and premature death. It provides novel evidence to support recommendations to modify eating behaviors for improving health. Even a modest reduction in sodium intake by adding less or no salt to food at the table is likely to result in substantial health benefits, especially when it is achieved in the general population, the researchers said. They concluded the obvious and evidence-based strategy with respect to preventing cardiovascular disease in individuals is early detection and treatment of hypertension, including lifestyle modifications, while salt reduction strategies at the societal level will lower population mean blood pressure levels, resulting in fewer people developing hypertension, needing treatment, and becoming sick. Not adding extra salt to food is unlikely to be harmful and could contribute to strategies to lower population blood pressure levels. How structure of antibodies is effective for cancer treatments. Researchers at the University of Southampton have gained unprecedented new insight into the key properties of an antibody needed to fight off cancer. The interdisciplinary study published in Science Immunology revealed how changing the flexibility of the antibody could stimulate a stronger immune response. The findings have enabled the Southampton team to design antibodies to activate important receptors on immune cells to fire them up and deliver more powerful anti-cancer effects. The scientists believe their findings could pave the way to improve antibody drugs that target cancer as well as other autoimmune diseases. In the study, the team investigated antibody drugs targeting the receptor CD40 for cancer treatment. Clinical development has been hampered by a lack of understanding of how to stimulate the receptors to the right level. The problem being that if antibodies are too active, they can become toxic. This latest research harnesses this property of the hinge and explains how it works. The researchers call this process disulfide switching. Researchers analyzed the effect of modifying the hinge and used a combination of biological activity assays, structural biology, and computational chemistry to study how disulfide switching alters antibody structure and activity. The approach was to analyze the structure of the antibody in atomic detail using the method of X-ray crystallography. While the resulting picture is very accurate, the information on how they move their arms is missing, and they needed an image of the antibody in solution, for which they used an X-ray scattering approach called SASS. They then used mathematical models and a chemical computing approach to analyze the data using the Southampton High Performance Computing Cluster IRIDIS, the researchers concluded. They also concluded that the study had given new information about how to engineer antibodies to deliver a better immune response. They proposed that more rigid antibodies enable the receptors to be bound closer together on the cell surface, promoting receptor clustering and stronger signaling for activity, paving path for cancer treatment. That's all for today. Stay tuned to Medical Dialogues for latest updates.
Never miss a medical update from Medical Dialogues. Like, subscribe, and press the bell icon.